All right, everybody, welcome to Kines 2168. My name is Joshua Tiller. I will be your TA this semester. I'm working on you throughout this lab. So first things first, I'm going to go through this first PowerPoint with the guys. First, I'm going to introduce the syllabus, and then we're going to start really going over some stuff we need for the first lab. So first, I just want to introduce myself. My name is Joshua Tiller. You can just call me Josh. I am a grad student here at UNCC, getting my degree in clinical exercise physiology. I did my undergrad at Wingate University, where I got a degree in exercise science, and I ran track there all 4 years. Um, yeah, that's me. So that's how I get started. So first, a real quick thing over the syllabus. Please read your syllabus. That's going to be key for your success in this class. All the information you're going to need to know regarding class is going to be in there. But some key highlights on what you guys to know was there's going to be something going to be 9 post-lab quizzes throughout the semester. Each going to be worth 10 points. There's going to be a dissection assignment we'll do later on this semester. That's going to be worth 50 points. Also later this semester, we'll have a physio exercise worth 30 points. There'll be 4 body viz assignments worth 20 points each. So right now I know body viz we're still working on how we're going to do body viz for this class, but as soon as I get that information, I'm going to get that to you guys as soon as I have it. And then there will also be 4 practical exams throughout the semester, and each exam is going to be worth 80 points each. Practical is going to be really important for this class. It's going to be your main exam for this class. I want you guys to do really well, so I'm gonna be working with you. As long as you work with me, okay? I'm gonna do my part. Make sure you guys know what you need, have what you need to know. I need you guys to do your part to make sure you know it for when it comes to exam. And also attendance is mandatory, and it'll be worth 30 points of your grade throughout this semester. That's the syllabus. I'll go over some more of that once we actually get into class. We'll look over that with you guys in person. There's also on Canvas, there is a word list for each practical section. Make sure you have to look over those because those will be on the practical. You'll be quizzed on those. So this first lab, we're just gonna go over an icebreaker, introduce myself. I'll try to get to know some of you guys once we get in person. I'll do a little tour of the mastering and body viz software. We'll go over the syllabus a little bit more, and then I will go through this PowerPoint and we'll get jump in the lab. As far as me, my office is in the Belk Gym on the second floor at the end of the hallway towards the right. My office hours will be Wednesday from 1 to 2 p.m. or by appointment if needed. So if Wednesday doesn't work for you or you want a little more extra time with me, shoot me an email and we'll get that set up. And then my email address is jtiller5@uncc.edu. I know some of you have already emailed me after I sent out that announcement late last week. Yeah, and as far as lectures go, going forward, I know this was coming late, like the day before class, but from now on, I would try to get them up and posted by Saturday afternoon, so that way you can have a couple of days to look at them before we get in the lab. And then I skipped over. So another thing, lab rules: closed-toed shoes are a must. So no sandals, no flip-flops, no Crocs. I need closed-toed shoes. If you don't have them, I will send you back to your dorm to go pick them up. This first lab will be a little more lenient on it because we're just trying to get stuff figured out. But going forward, starting next week, you must have closed-toed shoes. Also, there is no food or drinks allowed in the lab. If you have a water bottle, make sure it can be closed. There'll be no open containers. Next, the last rule is there will be no cell phones while in the lab because we don't want you guys taking pictures of the slides that we're using and the microscopes. You can bring your laptop and tablet and have that with you at your desk for notes and whatever that you need. For notes, body viz, MATLAB and Lasting, you can use laptop and tablets, just no phones. Here's some more laboratory safety regulations. I'll leave this on the screen here for a second so you guys can read through those. But I will discuss these in depth when we get in the lab tomorrow. All right, so first up, we're gonna go ahead and jump right into some lab stuff. So first exercise, we're going to go over the anatomical position and anatomical direction. So let's start up. Anatomical position is a really important thing you guys know early on. because it is a universal point of reference that we will use throughout the rest of the semester when trying to describe where something is located. So the anatomical position is going to be an upright posture. Your feet are going to be slightly apart, toes facing forward. Arms going to be down by your side, palms facing forward, thumbs pointing away from you. And then your head's going to be in a neutral position, eyes facing forward. So another really important thing as you see on this slide is on the top right, it says reference only. That is for the various regions that we see here labeled on this figure. So we go from top to bottom, front and back. It'll show you the labels of the different regions that you're going to need to know throughout this lab. So this particular slide is going to be a really good thing for you to study repeatedly and constantly, so that way you know where everything is whenever we say something in lab. So for example, if I say, show me where the pollex region is. If you don't know the pollex means thumb, then you're not going to know where that is on a practical. So make sure you guys know each of these regions. This particular slide exactly won't be part of a practical or exam, but you will need to know each region that is labeled on here. That's why it's listed as reference. You'll see shortly, another slide will say know everything. That means you need to know exactly everything on that slide as it is, like we see here. So on this slide, we have our anatomical direction. You see in the top right, it says know everything. That means this slide will be on some sort of quiz or practical, exactly like this. You need to know everything on this slide. So we'll start with this. We're gonna go from top to bottom, left to right. So you see the top left, you have your superior and inferior direction. So superior is going to mean above. Inferior is going to mean below. You can see here in the human figure, superior is more reference to the head or cephalic region, and inferior is going to be towards the feet. But you see in the dog, superior is going to be towards the dorsal or the back of the dog, and inferior is going to be ventral. Okay, so a really good way to think about this is superior is going to just mean above. Anything above is going to be superior, inferior is below. Next, we have anterior and posterior. So anterior is going to mean the front side of the figure, and posterior is going to mean the back side. And then if you look at the dog, you see anterior means towards the head, and posterior means towards the back. Okay, so that's one key difference you're going to need to know when you have a human or an animal figure, is you need to know what position they're in and what correlates to which one. Okay, so next we're going to go over two terms that are not on this slide, but you do need to know them. So next we have medial and lateral. Medial is going to mean close to the midline of the body. The midline is the line going straight up the middle of your body, through your head, down to your feet. So think midline, think sternum, belly button, nose. Those are going to be key points of midline. Lateral is going to be away from the midline, which is going to be away from the middle. So think fingertips, hold your arms out to your side. Your fingertips are going to be lateral to your shoulder. They're going to be further away from the midline than your shoulder. So next, we're going to have proximal and distal, and these are going to be in reference to your trunk or your torso. So you see in the figure, you have the hip is proximal, and then the feet are distal. That is because the hip is closer to the trunk or your torso of your body than your feet. Another example, let's go back to the fingers in shoulder again. You hold your arm up to the side. Your fingers are going to be distal because they're going to be away from your torso. Your shoulder is going to be proximal because it's closer to the torso. And then the last two things you need to know are also not on the slide, and that is superficial and deep. So superficial is going to mean towards the body surface, and deep is going to be away from the body surface. The easy example for this is going to be your skin and your bone. Your skin is going to be the most superficial thing on your body. It's on the top of the body surface. Bones is going to be one of the deepest things in your body surface. So think skin is superficial to the bone. Bone is deep toward the skin. So it's a little bit practice. So this is when those regions are going to need to be known. The regions on the previous slide where I said you need to study those, this is one of those things you're going to need to study. So, for example, the popliteal region is blank. The popliteal region is going to be posterior because the popliteal means the back of the knee. The popliteal region is behind the knee, which means it's going to be on the posterior portion of the body. The acromial region is blank to the otic region. So the acromial region is going to be near the shoulder, and the otic region is going to be your eyes. So that means your acromial region is going to be inferior to the otic region. The femoral region is blank to the tarsal region. The femoral region is going to refer to your femur or your thigh, and the tarsal region is going to mean your ankle. So that means the femoral region is superior to the tarsal region. That means because your femur is above your ankle. And again, this is one of the easy examples you're going to find. The bones are deep to the skin. That's just because bones lie beneath the skin. They're going to be deeper. They're going to be away from the body surface. And the skin is going to be the body surface. Next, we have our body planes. So there's three. There's the median or mid-sagittal plane. There's the frontal or coronal plane. And then there's the transverse plane. So the median or mid-sagittal plane is going to split the body into left and right sides. That's really the easiest way you can get it. If you want to think about, if you're an exercise guy like me, one thing I know is I can do median movement like a curl. I can do them on either side of the body and not have one interfere with the other. The frontal plane or coronal plane is going to split the body into front and back, or anterior and posterior regions. So that's going to split you straight down the middle. You're going to get a nice section. You're going to see the front and back side. So if you want to exercise like this, that's going to be like a lateral raise. You're going to go lift your shoulders up to the side. And then the third and final plane is the transverse plane. That's going to cut you in the middle from top to bottom or superior inferior regions. If you look below, you can see what each cross section would look like in that plane. So from the left, you have your median or mid-sagittal plane, where you would see half of the body on the left and right side. Frontal and coronal plane in the middle, you see the front and back of the body. And then the third one, you have your top and bottom. So this image is looking at the body cut in half, you're looking up. You can see the liver, the heart, the pancreas, and the spleen. So this is just another example of what each cross section would look like with each plane. So if you cut a banana and cut it in half, like you would say if you're chopping up a banana for a smoothie, you have a transverse cross section. This is what it would look like right here. B, if you cut banana down the middle a long ways, that is going to be your median section. It's going to get you a right and left side. And then you look at C, you have your frontal section. You're going to have three cuts for this banana. You have one that's in the back, you have one in the middle, and you have one so you can see the stem and the bottom of the banana. All right, so next exercise, we're going to go over the microscope. I'm going to breeze past these because this is something you need to know. You're just going to study this diagram I'm going to show you here in a second. So here is our microscope. We have our ocular lens, our rotating nose piece, our stage, our three objective lenses, which we'll go over a little bit more here in a second. We have our condenser, the iris diaphragm lever, stage light, the base, the fine adjustment knob, the coarse adjustment knob, the condenser knob, the mechanical stage, and the arm. You just need to really just look over this diagram a couple of times. It's not that hard, but you do need to know everything on this slide. You will be on a practical in the future, so make sure you guys go over this really, really well. And then use our three different lenses or all have different magnifications. So the blue one is going to be a 4 times magnification. The yellow one is going to be a 10 times magnification, and the red one is going to be a 40 times magnification. It's important to know, and I'll go back to the slide, that our ocular lens has a base magnification of 10. So when you're trying to calculate the number, like the amount of magnification that you're looking at a slide through the microscope are, you're going to take the number that corresponds to the color of the lens, multiply that by 10. So if you're using the blue lens, your total magnification is going to be 40 times. You're going to use the yellow lens, total magnification is going to be 100 because you do 10 times 10. And if you're using the red, total magnification is going to be 440 times 10. All right, so that was the microscope. That's everything you need to know for that. Make sure you guys really study that PowerPoint with the microscope diagram. And next, we're going to go into the really heavy stuff that's really important for this lab and the practical. So exercise 6, we're going to be looking at the 4 primary tissue types and the major subcategories of each type. For this, you're going to need to know where the types are, what their function is, and how they look. We're going to go over each one, each of their special characteristics, and their key functions. So the structural levels of organization, these are something that you're going to know. It's not that important, but you should at least know it. So from the most basic level of organization, you have chemicals, organelles, cells, tissues is what we're going to focus on today. Organs, which is just a collection of tissues, an organ system, which is a collection of organs. There's another one that is not listed on this slide that I think you should know is organisms. Organism is a collection of organ systems working together. All right, so first up, if we have 4 main tissue types, that's epithelial, connective, nervous, and muscle. So for these slides, we're going to go really heavy into the epithelial and connective tissues, and then nervous and muscle tissues we'll do toward the end. They're a little bit more easier, a little more straightforward for you guys to know, but really hammer home epithelial and connective tissues today. So first up, epithelial tissues. So epithelial tissue, really main functions are going to be protection, absorption, filtration, secretion, and transportation. Epithelial tissue is going to form the coverings of internal and external surfaces. They're going to line body cavities and hollow organs, and they're going to regulate the exchange of material from the internal and external environment. So some of the key distinct characteristics of epithelium is polarity. So it's going to be a free surface, which is going to be your apical surface, and it's going to be a connected surface, which is connected to the basement membrane. They're going to have specialized contacts because epithelial tissue is going to be tightly packed, but they're going to have special junctions to allow transport, such as gap junctions. They're going to be supported by connective tissue because they're all going to be connected to a basement membrane. That's one of the key things you can look for when you're looking at a slide to see if this is a connective or epithelial tissue. Epithelial tissue is going to have that basement membrane. And then also epithelial tissue is going to be avascular, so there's going to be no veins or arteries running through epithelial tissue to supply nutrients, but they are innervated, so they will have a nerve innervation. So they're going to rely on nutrients being diffused through the basement membrane, and those epithelial tissues that are greatly well nourished will be able to regenerate. So then there's 2 classifications of epithelium. You have they're going to classify by 2 things: the number of layers and the cell type. So this is going to be your number of layers. So you have simple and stratified. Simple epithelial tissue is going to be a single layer cell connected to the basement membrane. You can see here on the left. And then, like I was saying before, you have your apical surface and your basal surface. Apical surface is going to be on the top of the cell, top of the tissue, not connected to nothing. The basal surface is going to be what's connected to the basement membrane. And then here on the right, you can see our stratified epithelial tissue. And it's characterized by multiple layers of cells. And then next criteria, we're going to break epithelial tissue into its cell shape. So on the left here, we have squamous cells. They're going to be flat and squished cells. In the middle, we have cuboidal cells. They're going to be shaped like a cube with the nuclei in the middle. And then we have columnar cells. They're going to be long and skinny cells with one nuclei towards the middle. All right, so here we're going to start going over the different types of epithelium. So we're going to remember they can be simple or stratified, and we have their shapes: squamous, cuboidal, or columnar. So for these slides coming up, you're going to need to know everything. You're going to need to know the description, the function, and the location, and what they look like. So first up, we have simple squamous epithelial. So the description of this is going to be a single layer of flattened cells with dish-shaped central nuclei and sparse cytoplasm. This is going to be the simplest form of epithelia. The main function of this is going to be allow materials to pass by diffusion and filtration, and will also serve as protection. They also might secrete some lubricating substances in certain locations. So the location of these is going to be in your kidney, in your lungs, the lining of the heart, blood vessels, and lymphatic vessels, and also lining of the central lining of the ventral body cavity. So if you look here on the right, we can see there is a lot of empty space here. This is the picture of the lung. This is where gas exchange is going to occur. So these cells are going to be really thin and really tightly packed because you don't want anything other than oxygen and carbon dioxide diffusing through these cells. Here's another picture of simple squamous epithelium. You see the arrow here is pointing to this thin line here. That is going to be what your simple squamous epithelium looks like on a microscope. There's also various types of cells here, but we're really focusing on this one in here. Here's another picture of your simple squamous epithelium. Same thing to the first as the other one. You're really looking for a thin line of cells that are really flat and have a central nuclei. Here's one last picture similar to the one of the air sac we saw in the beginning. This is just what the air sac is going to look like on a microscope. So next up, we have our simple cuboidal epithelium. So the description is going to be a single layer of cube-like cells with a large spherical central nuclei. Their main function is going to be secretion and absorption. You're going to find these in your kidney, the ducts and secretory portions of small glands, and the surface of the ovary. So they're going to be characterized by tube-like shapes, going to be kind of equal on all sides, in a single layer. And you're going to also have that basement membrane here. And they're often going to open up into a duct. You're going to see a circle of cells and then an empty space in the middle, like right here. This is a picture of the kidney, of kidney tubules. Next up, we see here another slide of simple cuboidal epithelium. This is what it's going to look like on your microscope when we get in the lab. You see here where this arrow is pointing, we have a single row of square-shaped cells with their basement membrane opening up into an empty space, which is going to be the duct, which is where they will secrete whatever substance that they are creating. Here's another one, similar to the last one. Single layer, cube shaped, one central located nuclei opening up into a empty space, which is going to be a duct. And one last one, we can really see how the ring of cells open up into that duct. Next up, we have simple columnar epithelium. So this is going to be your one layer cell, long and skinny. When you have most of the nuclei going to be closer to the bottom of the cell. So the description we're going to go over this one is single layer of tall cells with a round to oval nuclei. One thing with simple columnar epithelium is that some will be ciliated. They will have little hairs that will be used to move things back and forth. They will often contain a mucus-secreting goblet cell. The function of these is going to be absorption, secretion of mucus, enzymes, and other substances. The ciliated type is going to propel mucus or reproductive cells by ciliary action. You're going to find the non-ciliated type. It's going to line most of your digestive tract from your stomach to your rectum. It's going to be in the gallbladder and excretory ducts of some glands. You're going to see the ciliated version lines the small bronchi, uterine tube, and some regions of the uterus. So we see here the arrows pointing towards the simple columnar epithelium. We have a single row of cells that are being long and skinny with a round nuclei toward the bottom, connected to a basement membrane of some sort. Next up, we have pseudostratified columnar cells. So this one's going to be a little tricky because you're going to want to think that it is a stratified type of tissue, but it is not. Pseudostratified columnar epithelium is going to be single layer cells of differing heights. Some will not reach the free surface, and then you're going to also see the nuclei on different levels. That's going to be the key thing that you know that is going to be pseudostratified, that the nuclei will appear to be on different levels. So you really need to make sure you pay attention and look to see if each nuclei is reaching the apical surface or the free surface, and if the nuclei are on different levels. Okay. And they will also contain those mucus-secreting goblet cells and have cilia. So the function of these is going to be secretion, mainly mucus, and then repulsion of mucus by ciliary reaction. So the non-ciliated type are going to be the male sperm carrying ducts and ducts of large glands, and then the ciliated variety lines the trachea and most of the upper respiratory tract. All right, so this is going to be our first stratified epithelium. We can have stratified squamous epithelium. So it's going to be a thick membrane composed of several layers. The basal cells are going to be cuboidal or columnar and metabolically active, which means they're going to be ones that reproduce. And your surface cells are going to be flattened squamous cells. In the keratinized type, the surface cells are going to be full of keratin and dense, so think of your skin. And then basal cells are active in mitosis, producing the cells of more superficial layers. So this is going to be mainly found in the linings of the esophagus, the mouth, the vagina, and you're going to have the keratinized variety form the epidermis of the skin, which is going to be a dry membrane. The function of this is going to be protection. So think skin. Your skin is many layers. You're losing skin flakes all the time, and you're being constantly reproduced. But your skin's main job is to protect you from the outside. So you can see here the different layers of the stratified squamous epithelium. Here's another picture here. Here we can see how on the right side we have that basal membrane with the cuboidal cells, and as you continue towards the left, towards the arrow, the cells get flattened out and becoming you see less nuclei because these are dying cells. All right, next up we have stratified cuboidal epithelium. So these are going to be generally 2 layers of tube-like cells. Their function is also going to be protection, and they're going to be in the largest ducts of sweat glands, mammary glands, and salivary glands. So you're going to find these really in your mouth and skin. So when you look here, we see our basement membrane, and we see 2 layers of cube-shaped cells. And then each cell has their own distinct nuclei. And they're going to open up into this duct lumen, which is where they're going to secrete either their sweat or saliva. Next, we have our stratified columnar epithelium. Do not get this confused with the pseudostratified, because I think you're going to tell the difference. With this one, you have several cell layers. Basal cell is going to be cuboidal, and then the superficial cell is going to be more elongated and columnar. Their main function is going to be protection and secretion. You're going to find these mainly in the male urethra and then some other small glands. But this is a pretty rare tissue type to find in the body, so it really is going to be in the male urethra. So you can see here we have our basement membrane, and then we have our row of cuboidal basal cells. And as you get above that, we have our long, elongated cuboidal cells. We have several rows of these. So next we have our transitional epithelium. So this is going to resemble both stratified squamous and stratified cuboidal. Your basal cell is going to be either cuboidal or columnar. And your surface cells are going to dome or are going to have a dome shape or squamous-like depending on the degree of the organ, how much it can stretch. So the function, this is going to be stretches vertically and permits the distension of urinary organ by containing urine. So it's going to be really found in the uterus, the urinary bladder, and parts of the urethra. So for this one, you're going to see a basement membrane, a row of cuboidal or columnar basal cells, and then as you go up, you're going to see mainly cuboidal or squamous cells. All right, so that was going to be all of our epithelial tissues. Really go through those a couple of times, make sure you guys got those. We'll look at some more in the lab. If you have any questions for me, when we get in the lab, I'll be happy to answer those. So next we have our connective tissues, and there's going to be 4 types of those. We have our connective tissue proper, cartilage, bone, and blood. So the general characteristics of connective tissue is going to be their function is going to be mainly protection, support, nutrient transport, and repair. Most connective tissue is well vascularized, and means you have a lot of blood vessels within them. There are going to be many different types of cells present, and most of the tissue is going to be a non-living material called the extracellular matrix. So look on this diagram here, we want to know everything on this. So you can see different types of cells. So we have our macrophages, fibroblasts, lymphocytes, fat cell, mast cell, neutrophils. We have capillaries, which are going to be our blood vessels. Neutrophil, mast cells, lymphocytes, and macrophages are going to be a specialized type of white blood cells. They're going to function in your immune system. And then on the other side of this chart, we see our extracellular matrix, which is going to be our ground substance, which is going to be this light blue liquid looking substance here. And then we have our various fibers. So we have our collagen fibers, which are going to be our thick and strong ones. It's going to be the light purple shaded fibers here. We have our elastic fibers, which can be very bendable. That's going to be the orange fibers here. And then our reticular fibers are going to be the really small, light blue ones. And those are going to be really strong, but really light and thin. So the first up on our connective tissue is the loose connective tissue areolar. So this is going to have a gel-like matrix with all 3 fiber types. You're going to see reticular, elastic, and collagen fibers all found in this type of tissue. You're also going to find fibroblasts, you're going to find macrophages, mast cells, and some other white blood cells. The main function of the loose connective tissue areolar is going to wrap and cushion organs. It's macrophages phagocytize bacteria, which means it's going to eat and destroy bacteria. It also plays an important role in inflammation and holds and conveys tissue fluid. Locations is going to be really all over the body. It's going to be widely distributed under all epithelia. It's going to form the laminar propria of mucous membranes. It's going to also package organs and surround capillaries. So if you look here on the right, we can see how you have all the bunch of different types of fibers going to be very loosely distributed throughout the tissue. And you're going to have your fibroblasts and their nuclei. So we have here the collagen fiber is going to be the really thick pink ones. Your elastic fiber is going to be a little thinner ones, and you're not really going to be able to see the reticular fibers because they are so thin. Here we have another example of our loose connective tissue areolar. You can see the very vastly dispersed fibers throughout the tissue. And here's what it's going to look like under the microscope. Look here, we have our fibroblast nuclei, our gel-like matrix, which is the lighter purple, and then the dark purple lines are going to be the 3 fiber types throughout the tissue. And here's one last example of what it can also look like under the microscope. And this one is a little harder to see the fibroblast nuclei, but if you look closely, you can see little dark dots throughout the tissue. And then, of course, you can see the vast network of fibers throughout the tissue. Next up, we have our adipose tissue, which is going to be another loose connective tissue. So adipose tissue is just fat tissue. So the matrix is going to be similar to areolar, but it's going to be very sparse. The cells are going to be closely packed adipocytes or fat cells. and their nuclei are going to be pushed to the side by the large fat deposit in the middle. So you see in the image, we see these kind of grid-like or honeycomb-like patterns. So that's going to be your adipocytes. You see the dark dots going to be the nuclei, and then the big open space in the middle is going to be your fat droplet within within the adipocyte. The function is really going to be provide reserve fuel, insulation against heat loss, and support and protect organs. The location is going to be under the skin, around kidneys and eyeballs, within your abdomen, and within the breast. So this is what it's going to look like on the microscope. As you can see, we have that, again, honeycomb-like structure here, the wide open spaces in the middle, which is going to be your large fat droplets within the adipocyte. And you can see the dark purple dots along the honeycomb structure, which is going to be the adipocyte nuclei. Next, we have another loose connective tissue. It's going to be reticular tissue. So the description of this is going to be a network of reticular fibers in a typical loose ground substance. And then you also have reticular cells that lie on the network. The function of this is going to be we're going to form a soft internal skeleton that's going to help support other cell types, including white blood cells, mast cells, and macrophages. So really the most often reticular fibers are going to be there to support other cells. That's one thing the kid to know. And they're often going to be blood cells or lymphocytic cells, like white blood cells, mast cells, and macrophages. So you're going to find these in your lymphoid organs, so you're going to find these in your lymph nodes, bone marrow, and spleen. There's going to be all organs that function within your immune system. So we see here, we see this dark network of reticular fibers and these small, round reticular cells connected to these fibers. And then you can also see here the smaller dark purple kind of bluish cells, maybe your white blood cells or your lymphocytes. They're going to be supported by this particular tissue. This is another image of reticular tissue on a microscope, but we won't use this image often in lab because it's not really what it's going to look like when we see it in person. It's a little bit of an older image to what they used to look like, but now that we use a new stain, we're also going to see them in this dark purple. So next up, we're going to move into our dense connective tissues. So the first one is going to be dense regular connective tissues. So the description is going to be a primary parallel collagen fibers, a few elastic fibers, and the major cell type is going to be fibroblast. And these are going to attach muscles to bones or bone to bone. So it's going to be found in your ligaments and your tendons. And they're going to be able to withstand the great tensile stresses when pulling forces applied by muscle. So you're going to see that they look really uniform in this nice, even pattern, kind of a wave-like pattern here. They're all going to be moving in one direction. That's because they're all going to be the way muscles work when they pull on the tendon. They're all going to pull in one direction, and you want your tendon to be able to transmit that force to the bone to be able to move your joint. And then next we have our elastic connective tissue, which is these dark elastic fibers in this kind of same uniform pattern. And the key difference between the dense regular connective tissue and elastic tissue is that elastic tissue is going to be this much darker, much more tightly packed. It's much harder to see the individual nuclei in the elastic tissue. So the description is going to be can contain a high proportion of elastic fibers. Their function is going to allow the recoil of tissues following stretching. We're going to maintain the pulsative flow of blood through arteries and aid the passive recoil of lungs following inspiration. So you're going to find these in the walls of large arteries within certain ligaments in the vertebral column, and they're going to also be within the walls of bronchial tubes. So next up, we have our dense irregular connective tissue. So you can see this is really different from the dense regular connective tissue and how in its form. So one thing that's going to be important to know for this class is that form follows function. So if we go back to our dense regular connective tissue, they're all running in one line. That is because they're transmitting forces in one direction. If we go back to our dense irregular connective tissue, you see that they're running in various different directions. That's because they're going to be pulled in various different directions. They need to be able to withstand that force. So the primary description, this is going to be a primary irregular arranged collagen fiber. There are going to be some elastic fibers, and the major cell is going to be fibroblast again. The function is going to be able to withstand tension exerted in many directions and provide structural strength. So again, you're going to see them running in multiple directions because they're going to be pulled in different directions. So it has to be able to withstand that force in many directions, unlike the dense connective tissue, which is going to be pulled in one direction. Occasionally, it's going to be finding these in your fibrous capsules of organs and joints, the dermis of the skin, and the submucosa of the digestive tract. So again, the key thing with these is going to be how it looks. You're going to see all the collagen fibers running in various different directions, and you're going to find nuclei, fibroblasts distributed throughout. Next up, we have cartilage. We have our first type of cartilage. It's going to be hyaline cartilage. For these, you're going to have your chondrocyte, and they're going to live in this empty space called lacunae. So these are going to have an amorphous but firm matrix. Collagen fibers are going to be dispersed throughout the network, but you're not able to see them directly. Chondroblast going to produce the matrix, and when they mature, they're going to lie in these empty spaces called lacunae. Their function is going to be support and reinforcement. They serve as a resilient cushion and they resist compressive stress. So you're going to find these in most of the embryonic skeleton. You're going to cover the ends of long bones and joint cavities. They're going to form your costal cartilage. At the end of the ribs is going to be the cartilage that your nose, your trachea, and your larynx is made up. So these can be very strong, dense cartilage. You think about it in your throat. If you were bend your neck, you don't want your throat to close. So it's going to be something that's going to be able to keep its shape without being too distorted. So this is another example of the hyaline cartilage on a microscope. You can see the chondrocytes, all the little dots throughout the extracellular matrix, and the open spaces are going to be the lacunae that the chondrocytes are going to live in. Here is another example of the hyaline cartilage on a microscope. You can see here this dark purple in the middle here is going to be your extracellular matrix, and then the darker, almost black dots are going to be the lacunae where the chondrocyte is going to be located. Next up, we have elastic cartilage. It's going to be very similar to hyaline cartilage, but it's going to have a more elastic fibers in its matrix instead of collagen. The main function is going to maintain the shape of a structure while allowing great flexibility. So think your ear. You can bend your ear all kinds of ways. It's going to bounce back into its original shape. It's also going to be in your epiglottis and your throat. So again, with the elastic cartilage, you're going to have a chondrocyte. It's going to produce the extracellular matrix, and it's going to be within an empty space called the lacunae. Here is another example of the elastic cartilage on a microscope. You can kind of see the difference between the elastic cartilage and the hyaline cartilage. Hyaline cartilage is going to be much more densely packed, while the elastic cartilage is going to have a lot more space in between to have a wider lacunae for the chondrocytes to stay in. And here is another example of the elastic cartilage on a microscope. Next up, we have fibrocartilage. This is going to be found in the intervertebral disc between your vertebrae and your spine, also in your pubic symphysis, which is the joint in between the two sides of your pubic bones, also in the disc of your knee. So the description, this is going to be a matrix similar to, but less firm than the matrix of hyaline cartilage, and you're going to find thick collagen fiber is going to be the dominant structure in this sort of tissue. On the function, this is going to be tensile strength with the ability to absorb compressive shock. So think in your spine, we want to cushion any kind of force that goes through your spine so that the vertebrae are not being pressed into each other. So if you look at the image here on the right, we have our network of collagen fibers going kind of in sort of in one direction. And then we have our chondrocytes still living in our lacunae. Um, and you're periodized by this blue and purplish shade for the fibroblast. So that's the end of our cartilage. So make sure you definitely go over those a couple of times. And then next we're gonna go into some more connective tissue, a little bit easier one. So we have bone up here next. It's gonna be a really distinct image. One of the things we're gonna look for is rings on the microscope. And you're gonna have a hard calcified matrix containing many collagen fibers. We're gonna have osteocytes living in the lacunae again, and they're going to be very well vascularized. The function of bone is going to be support and protection. It's also going to provide levers for the muscles to act on. It's going to store calcium and other minerals, and fat. And then the marrow inside bones is the site for blood cell formation. Location of bone tissue or osseous tissue is going to be in the bone. So again, really make sure for the thing for bones is you look for these rings you see here around the lacunae in the middle. It's going to be a central canal. It's going to be where the blood vessels flow. And then these little dots around the outside are going to be your osteocytes and within the lacunae. And then the space in between the different lacunae and osteocytes is going to be called the lamella. Here is another image of a bone tissue on a microscope. You can still see here we have these really distinct rings of lacunae and a central canal for the blood vessels to flow through. And here is one last image, a little more zoomed out, but you can still clearly see the central canal for the blood vessels to flow through and the rings of lacunae breaking down from within there. Next up, we have blood. Blood is going to look like a bunch of red dots, red circle dots, and you have our plasma matrix here. So yeah, description, you're going to have red and white blood cells in a fluid matrix. That fluid matrix is going to be your blood plasma. The function is going to be transport of respiratory gases, nutrients, and waste, other substances. And these are going to be found within blood vessels. So this one's really easy to see. I'm kind of going to brush past this one because it's pretty simple in my opinion. It's just going to look like a bunch of red dots or stuck on each other. And then you have these other white blood cells, which can be lymphocytes and neutrophils. Here's what blood looks like on a microscope. It's really distinctive, guys, so don't spend too much time on blood. Really focus on your epithelium and your connective tissues. But as you see here, blood looks like a lot of little tiny dots within a loosely packed plasma matrix. So next up, we have our last two types of tissues. We have our, not last two types of tissue, last tissue type. We have our nervous tissue. They're going to be found in two places. It's going to be found in your central nervous system, which is going to be your brain and your spinal cord, and your peripheral nervous system, which is going to be all the nerves that run out from your spinal cord into your extremities. There will be two major components. You have your neuron and your supporting cells, your neuroglia. And the primary function is going to be communication. So they're going to be, in the key characteristics of nervous tissue is going to be that they are excitable and that they are conductive. So here is nervous tissue. The description of nervous tissue is going to be that you have neurons that are going to be branching cells, cell processes that will be quite long, extended from the nucleus containing cell body. Also contributing to nervous tissue are non-excitable supporting cells, which we talked about before. So the function of nervous tissue is going to be transmit electrical signals from sensory receptors to the effectors, which will be muscles or glands, and then supporting cells also help protect neurons. Location is going to be brain, spinal cord, and nerves. So if we look at the image here, we have our main cell body. Coming off of the cell body, we have our axon, which is going to be covered by a myelin sheath. And you can't really see them in this image very well, but also coming from the cell body, you're going to have little dendrites, it's going to be little smaller branches, similar to the axon. And then you also see here the little dots in between the axons and connected branches are going to be our supporting cells. And again, here is another image of nervous tissue. We see our darkly stained cell bodies You can kind of see the matrix of axons and dendrites as they kind of start to connect. And then you see all the little dots are going to be the nuclei of our supporting cells. Next up, we have muscle tissue. So muscle tissue is going to contain contractile protein fibers allowing for the shortening of the cell, which is going to result in movement. So we have gross movement, heart contraction, and then digestive motility. Gross movement is going to be your skeletal muscle. Heart contraction is going to be your cardiac muscle, and digestive mobility is going to be your smooth muscle. So first up, we have skeletal muscle. It's going to be characterized by these long cylindrical multinucleated cells with obvious striations. You can see here in the image that there are obvious stripes going in one direction for a skeletal muscle. That's going to be because skeletal muscle is only going to be moving in one direction. Everything's going to pull in line with each other to create force on the bone to make the joint move. Function is going to be voluntary movement. So this is going to be something that you can control. You can control when you want to flex your bicep or when you want to lift your arm. So it's going to help you with locomotion, moving things in the environment, facial expressions, and other voluntary control things, like throwing the ball. We're going to find these, and skeletal muscle is going to attach to bones or sometimes the skin. And we see here, you can see the striations within the muscle are multinucleated group of cells, and they're all going to be moving in one direction, working together. Next up, we have cardiac muscle. Well, these are also going to be striated. They're not going to be in a single uniform line. Cardiac muscle cells or cardiac muscle tissue is going to be branched. So we're going to have a branching, striated, and uninucleated cells that are interdigitated at special junctions called intercalated disc. So the intercalated disc is going to be what allows the different cells within the tissue to talk to each other. You can see them by these dark bands in between different cells. And again, you can see that the cardiac tissue is going to be branched, unlike the skeletal muscle tissue. And while cardiac muscle is also striated, it is under involuntary control. It would be a really bad thing if you get to think about your heart beating 24/7. It's something you kind of want to take care of its own. So its function as it contracts, cardiac muscle is going to propel blood in the circulatory system, and it's going to be found in the walls of the heart. Here is another image of cardiac muscle tissue on a microscope. You can kind of see here how you have these long branching cells with a single nucleus attached to each cell, and you can see the dark bands going across each cell, which is going to be your intercalated disc. And here is another image of the branching you will find in cardiac tissue. All right, last up, we have smooth muscle tissue. You'll see how this one looks very different from the other two because there are going to be no striations. So smooth muscle tissue is going to be spindle-shaped cells with central nuclei, no striations, and cells going to be arranged closely in form of sheets. The main function of smooth muscle is going to be to propel substances such as food or urine or a baby along internal passages. It's going to be under involuntary control. I mean, you cannot control these muscle tissues or muscle cells. It's going to be mainly found in the hollow walls of organs. You can see here we have these sheets of cells with a single nuclei, and they're going to be all tightly packed, forming this tissue. All right, so that is all of our cells, our tissues, and all right, guys, so that's going to be the end of our presentation today. That's going to be covering all of our tissue types. I really recommend you going through them a lot, especially the differences in the connective tissues and epithelial tissue, because there's going to be really important things for you to know. So on this slide, I have highlighted all of the tissues that you're going to need to know for the practical. The ones highlighted are going to be ones you need to see through a microscope. And the ones with the star are going to be not on a microscope, but you're going to need to know them from images from the slides. So then post-lab quiz will be due next Friday, not this Friday. We're going to give you guys an extra week to get this one done. So it's going to be due on September 2nd, I believe. Make sure you do that before it closes, guys. Remember, these are open book, but I very highly recommend you study first and then do the quiz. That way you can work on make sure that you know what you're looking at. All right, so that's going to be the end of my presentation, guys. I look forward to seeing you in class tomorrow. And once again, I know this one's coming out kind of late. If you don't get to it before lab tomorrow, that's fine. I will go over this in class. But and then going forward, I would try to have all of these things recorded and posted for you guys by Saturday afternoon. All right, that's it for me. I'll see you guys in class tomorrow.